Preti. Actually, Dr. Fennessey and I graduated from the same group in MIT's chemistry department. So we're both chemists by training, and but I currently am at the Monell Chemical Senses Center, where I've been for the last almost 42 years. And my primary research interest has been the nature and abundance of odors produced by humans and what kind of odorants emanate from the human body. And we also look at their biogenesis, that is where they come from. So that in brief is what me and my research group does. Danny?
Hi,
Danielle?
my name is Danielle Reed, and I'm a geneticist, and I work closely with Judge Preti on the TMAU project. And I'll tell you more about my research at the end of the talk.
And I'm Paul Fennessey. I'm, as you heard from George, a chemist also. I joined the University of Colorado, Denver, Anschutz campus in 1974, and we found our first TMA patient in 1977 or 8, somewhere in that time frame. And we've been addicted to TMA research since. So I think I'll start the process here. I want to really start out by saying that I probably would not have stayed and gotten involved with TMAU except for having Susan Joa at my side. Susan worked with us, and I think most of the patients that were identified in the first few years of our project, they thought Susan was running the project, which in fact she was. And I just want to make note that she was a dedicated person, and if she were here today, would be anxious, jumping up and down, wanting to talk more about TMA patients and how to take care of them. So here's an outline of our presentation. You can see that I'm going to talk about the disease entities. Dr. Preti will talk about evaluation of malodor problems, and Dr. Reed will talk about genetics of this disease. So this is the portion, my portion, recognition of TMAU as a disease. And I started out with some sort of eclectic slides. This business of public empathy, I work at a children's hospital, and every time I get depressed or unhappy with what's going on, all I need to do is walk out in the hallway and come across a number of children who have horrendous physical, mental, and or other kinds of problems, and it usually brings me out of my malaise and gets me to start to think about how minor my problem is. Unfortunately, that kind of empathy doesn't exist very well for patients with TMA excess. And you know, all you have to think about is being in a room or an elevator with someone who has a bad odor, and instead of really feeling sorry for them and trying to understand them, I think most of us, our gut reaction is, ugh, why didn't they take a bath? What's wrong with this person? And as a result, I think most TMA patients suffer from the social isolation caused by those of us who don't understand them and don't understand their disease. And oftentimes, and really depending on each person's personality, this can lead to isolation and I think some traumatic problems that need to be dealt with on a psychiatric level, not on a biochemical level. And it's really just an unfortunate sort of side effect of being affected with TMA excess. So the other issues that come around with this disease are the medical schools don't really understand or train patients to understand the sort of the odor caused by TMAU, the genetics of the disease, how to deal with it. And it's only because we've really identified it as a true rare disease, if you will, in the last 15 or 20 years, and a number of the textbooks take a long time to sort of get to a point where they're actually putting case reports in those textbooks for medical students and nurses to learn about. So there's a lot of ignorance, I think is the right term, about this disorder. The other things that make it difficult are the episodic intensity of the odor. And I'll talk a little bit about this later on, but since people don't always smell the way as intense as, very intense maybe, or that's a variable intensity, it makes it difficult for doctors, nurses, health workers in general to actually smell them and what kind of odor they have with them, and really identify it. And then finally, the genetic tests that we are doing now, and I think Dr. Reed will talk about this later, it's often really unclear and inconclusive unless you really have a clear picture of the patient and their family and their history. So my final note on this medical ignorance thing is really, thank goodness for the internet, thank goodness for programs like this one, and for patient support groups, because I think these are the major sources today of both healthcare workers, physicians, as well as nurses and others, to get information about trimethylamine, and for patients to begin to understand this disease and how you deal with it and what's causing it. And I think when you understand that, it makes it so much easier to begin to deal with how to handle it, how to address it, and how to explain it, I guess is the right term, to your peers and to your employers and your friends. So just from a historical point of view, there was a couple of reports in the Lancet from 1842 and 1858 that suggest that they had identified patients who had trimethylaminuria or trimethylamine excess. But the first actual clinical description and identification of the culprit, namely TMAU, trimethylamine, was done by a group in Denver, and it was a patient who was in the hospital for other genetic problems and some cardiovascular issues, but the laboratory was smart enough to be able to collect urine, collect saliva, and be able to identify trimethylamine as the cause of the odor. And from that point on, there have been a number of reports isolated around the country from various laboratories. Our own, Dr. Preti's location the Monell group has done quite a few. There's been a number in Arkansas and other places, all trying to understand what this disorder was. And there was initially some data in that group of reports suggesting that diet was one of the had an important role in this disease. And it wasn't until in the 80s that the genetics of this disorder began to be uncovered. And then that's continued from that point on, and Dr. Reed will deal with that as her part of the presentation. But it's now just been probably the last 15 or 20 years that this disease has really been recognized as a true inborn error of metabolism, if you want to call it that, or genetic disorder, and people have begun to understand in much more detail how to deal with this problem. So when we first started this project in 1978 or 79, 78, something like that, our method for measuring trimethylamine was just to measure the absolute concentration of trimethylamine in the urine. And it worked, but it was very erratic. And in looking at the literature and some of the other studies, it became very clear to us that the concentration we would measure was dependent upon the diet of the person who we were testing and the food they had eaten prior to our test. And so in 1984, we decided to moved to a choline test load to try to make sure that the patients had adequate amount of precursor in their diet to be able to sort of pick those patients who had to where the concentration of TMA would have much more meaning, and to look at the ratio of TMA to its metabolic product, the trimethylamine oxide. And looking at this ratios, we were able to make some much more definitive diagnoses, and it's been the basis for the biochemical testing that we've done ever since. And in this test load, we put in about 20 to 25 times the recommended daily dose of choline. So it's not a huge amount, but it is enough to drive the production of trimethylamine. And I'd say there are, as a footnote to this, those of you who are listening to this who either think you have trimethylamine or have patients that you think might have trimethylamine, there are lots of causes of human odor, and you shouldn't make any assumptions. I think since there is a biochemical test, you should do that before you start to take any other steps, I guess, to deal with this problem. And George will get into that as he goes through his portion of the presentation. So just as a way of substantiating in our own mind that choline played a role in this disorder, we had a second case in Denver of a 7-year-old boy, actually two brothers who both had trimethylamine, and they worked very closely with the dietitian at Children's Hospital to actually titrate the amount of choline down. And you can see from this slide that the dietary choline intake was decreased from about 500 milligrams a day to 231 milligrams after 4 weeks, which is very close to a minimum requirement, and why we wanted to have a dietitian working with them to do this. And we did all kinds of biochemical tests for liver function and other kinds of blood tests, and really couldn't see any deviation from normal. But the good news was this young man really had no detectable odor when his choline intake was reduced to 260 grams, 250 grams. And the TMA concentration in the urine was down. And then at the bottom you'll see we did a correlation between 24-hour choline intake and 24-hour urine TMA concentrations, and found that this had I'll move my little pointer down if I can get there. Anyway, it had a very good correlation with the choline intake and the TMA production. So then, as an example, and just as a way of a note that not always do these methods work, we tried a 67-year-old patient that we had to reduce his choline. It was from 1,000 or 1 gram a day to about half a gram a day, but he remained symptomatic. Namely, he continued to have an odor. And I think mostly for those of us who are mature, we have already picked the kinds of foods that we want to eat, and he wasn't very interested in modifying his diet down to the levels that would have brought him, I think, down to the minimum requirements, which is more like 300 or 400 milligrams a day. And you'll see on the next slide that the National Academy of Science has estimated, I think is the right term, these levels. And as you can see for children, for our first child that we did the study on, the 4 to 8-year-old level was 250 milligrams a day as the recommended adequate choline intake, and we were right at that limit, and I think we were successful there, whereas with this older man we were still at 900 and the recommended levels are 500. So it's really quite a variability in both the individual and the genetic problem that they happen to have, which enzymes are missing or not missing, or whether they're partially active or not, that actually controls whether you can manage the diet using a choline restriction. So then the variability issue comes up, and this was one that was of really interest to us because we wanted to know if you took this choline dose, how long did it take for trimethylamine to show up in the urine? And as you can see here, for the average, if you want to call it that, it was an 18 to 16 hour delay period from the time they took the choline load until the maximum amount of trimethylamine was detected in their urine, which means that if you're presenting yourself, or if you're a physician's office, for example, presenting yourself with trimethylamine and having them try to evaluate it, what you smell, what they smell, is really a result of what you ate yesterday, not anything else. So if you yesterday had a very low level of choline, you might not smell at all, and if you had a excessive amount of choline, you could smell pretty bad. And I think in the cases of these oral loads, this 5-gram oral choline load, most of our subjects had to be alone, I think, or be isolated during the 24-hour period after they took this load, just because of the intensity of the odor. And the other piece of evidence that supports the oral choline control was a report in the literature in about 1982 or 3, in which the patients were given antibiotics for a 2-week period, and these antibiotics were intended, they're the same ones used by the surgeons to sort of clear your gut of bacteria if you're going to have GI surgery. The bacteria is thought to be reduced by like 90% by the antibiotics, and when they did this, their patients with this 2-week load of antibiotic, they were the odor disappeared, which again suggests that it's the bacteria in the gut that's the main culprit. And this is just our guesstimate of what those differences are. The body has to have a whole series of compounds in it that have trimethylamine as a component of the biochemicals. And I think Dr. Reed will talk about that in some cases for small molecules that all end up producing trimethylamine. And these are intrinsic, and one has to have them, which is why there is a minimum daily requirement for various components. But it's our guesstimate that the turnover and the production of trimethylamine in the body is really in the order of 0.1 to 0.001 milligrams, so a very, very low level of compound that's turning over at any period of time. Whereas if you look at the estimated production of trimethylamine from gut bacteria, where we're taking in essentially hundreds of milligrams of choline, you're getting production of at least 1 milligram, if not more, so that the difference between the intrinsic production and the production of trimethylamine caused by the diet is almost 1,000 times different, which is a major, should I say, sort of defense or argument as to why diet plays such a major role in this disorder. So, from a summary point of view, the information about this disorder really needs to be taught in a much more broad way to healthcare providers, and for those of us who have now access to the net, providing people with the webinars that are being produced today and the earlier ones is key to helping the poor internal medicine physicians who are trying to diagnose these patients figure out what this disorder is and how to deal with it. There is a biochemical confirmation of trimethylamine, and it's critical that patients have this test, and George will, I'm sure, elaborate on that. And then Most patients don't have affected parents or siblings, and so from a genetic point of view, it's often hard to pin down that this is a genetic disorder, and I'm sure we'll hear more from Dr. Reed about that aspect. And so with this, I would turn over the microphone
Thank you,
to
Paul.
Dr. Preti and allow him
Okay.
to talk about individuals with malodor.
I generally start my talks off talking about human body odor, normal body odors. I often have a friend illustrate this, my friend the caveman, who's very hairy and no doubt was very smelly for his time. Normally, most human odors are formed by interactions of skin gland secretions and the microorganisms that live on our body. So you may have noticed that all different parts of your body smell differently, and that is because, and I've listed most of the odor-producing areas here, and the reason for all these smelling differently is that they all have different populations of bacteria, and they all have different types of skin secretions and available oxygen. And certainly deodorants and antiperspirants and widespread use of consumer products and fragrance soaps have been around for about 100 and some odd years. So before this, it was much more evident that humans emitted a variety of volatile odorous chemicals that caused them to smell. And I'm sure back in the Old West and pioneer days that people smelled a lot more than they do now, and you had many more odors present at a social distance, and that's a very critical thing I'll talk about: social distance versus intimate distances for odor production. So because of my expertise in body odor chemistry, we began to have many people referred to us, both by clinicians and dentists, or physicians and dentists, people who claim to have odor production problems but checked out clinically to be fine. So now with the advent of the internet, many are self-referred. These people came to us from all over the country, and some from other parts of the world. And as I said previously, many have been seen by at least 4 or more clinical or dental specialists in a quest to determine what makes me smell bad. And because of this influx of people, we developed a test or a protocol, a clinical workup, if you will, and I call it my analytical and sensory workup of patients. The critical part of which is a Choline challenge test to determine whether or not the patient has trimethylaminuria. So I'm not a real doctor, I'm a PhD, I'm not an MD, but we do see a clinical population and we do try to evaluate them for odor production. And part of this evaluation is the Choline challenge test, and we will sniff the patient. We will walk around, my technician and postdocs and I will sniff you, the upper part of your body. We don't get, we only do non-intimate parts. And we will tell you if you have an odor production at social and intimate distances, and how bad that is and what it smells like. The Choline challenge test actually came about when Paul and I went to graduate school together, and we bumped into each other at a meeting. I think it was a mass spectrometry meeting in Miami in 1989. And Paul told me they had developed this test, and we had just seen our first patient, and I had sent him urine from this patient, and then we began, when he told me that this test was now in place, we began administering it to everybody that came in. So it's become a staple in the diagnosis of patients. And this Choline challenge test has revealed the large undiagnosed population of individuals with TMAU within the group of people who are brought to us. You've already heard most of these facts, that trimethylamine is a gas at body temperature and has a strong, pungent, offensive, fishy odor. Excuse me, at lower concentrations, it may just be perceived as foul or unpleasant. And it has been reported to be found in all body fluids in normal individuals as well as people with TMAU. Trimethylaminuria, because of the strong fish odor, has been come to be known as the fish odor syndrome. I argue that this should not be used, since only about 10% of the people we have seen who have been diagnosed with TMAU have a severe form that causes overt fish odor production. And when I talk about severe or mild cases, I'm talking about the type of genetics that are underlying the presentation of the disease. And as we said, Danny will deal with these in the last part of the talk. As Paul rightly noted, the symptoms are sporadic and may escape notice due to low choline intake. And there's also what's called a specific anosmia for trimethylamine. That is, a certain part of the population, a very low percentage, maybe 3 to 5% of the population, lacks the olfactory receptors to actually perceive the odor of trimethylamine. So it may escape detection either because you're not producing a lot or because those around you may have this very unusual specific anosmia. We have seen in excess of 300 people in person, 350 people in person, and that number is certainly climbing. And of that, about 33%, about a third, have been diagnosed with some form of TMAU. All the data I'm going to present here has been published in a summary paper on the demographics and diagnosis of TMAU in people with what's called idiopathic malodor production. So there is a manuscript, peer-reviewed manuscript in the medical literature that defines this. Anytime a physician writes to me talking to me about diagnosing a patient, I send him or her the paper that we have published so they will become more educated, and in the hopes of relieving this ignorance barrier that Paul has talked about. So the average age of our TMAU affected population in this population that we've seen is 43.6 years, and there's a wide range, as you can see. Those who test negative for TMAU also have about the same average age. This is a very critical slide, I think, because in the medical literature, when you read about TMAU, almost certainly the description of the patient within the introduction of the manuscript says that the patient presents, the patient smells like fish. Or I can tell just from reading that the person writing the paper has not, probably has never seen a TMAU patient in person, but has just accepted as working with their blood or an isolated sample from the patient. These are in the words of the patients who come to me. These are how they describe their own body odor. I ask them to give me a sentence or tell me what part of your body, or tell me why you have come to me, what part of your body smells, in your own words. And this is the breakdown of symptoms we have seen. Unlike this one here, the classic, the second body odor is probably the classic presentation. People say they have a body odor problem. But you can see a great many people have a bad breath or odor. They complain of oral malodor, they complain of genital odor, malodor, or they complain of body and oral and genital things all together going on. So it is difficult for a physician, if taking a history, to say just from from a history of symptoms how the patient, whether the patient is TMAU positive or not. We have 3 times more women than men that are TMAU positive, and many of these women are women of color. So if you look at the next slide, a slide here, on the left-hand side you see that the purplish color or the reddish color is how many women of color we would expect to have in our population that are TMAU positive. And the blue color represents the number of women we actually diagnose. On the right-hand side are the Caucasian females. That's about the observed and expected populations are about the same. But we're on the East Coast. We're in a large metropolitan area. Many of our subjects are African American. if you did this on the West Coast, and I think I've seen data from John Cashman's group when he was still doing diagnosis, which suggested that he had a large population of female Hispanic women. So this may be population dependent, but regardless, it looks like more women either have this disorder or are more readily come in to a physician or to me complaining about the symptoms that they have. Also, women are affected depending on the part of the menstrual cycle they're in, and I think Dr. Reed may talk about some of this later. But women within probably, if we focused in on the right-hand side of the slide, as you get closer and closer to menses, your ability to oxidize trimethylamine to trimethylamine oxide, percent TMAO. As percent TMAO goes up, you become less symptomatic. As it goes down, you become more symptomatic. So women during the time of menses are more likely to have symptoms than during non-menses times. And that is because of there is a sex steroid effect on the efficacy of the enzyme that is responsible for oxidation. There's been many things that have been associated with TMAU particularly in the early literature. And even now, I often get people asking me lots and lots of questions regarding, if I have this, will I get that? And because I have TMAU, will I get this? Or because I have such and such a disorder, does this mean I have TMAU also? Well, none of these things have really, there has been no big study to demonstrate a relationship between all of these other problems that have been found in certain TMAU patients with the actual trimethylaminuria. But some of the genetics work that Danny is now doing, which she'll talk about, the Exome study, may show relationships between some of these other problems that patients have and the actual trimethylaminuria. The one in the middle, the psychiatric dysfunction, I'm not saying that people with TMAU have a psychiatric dysfunction, but many people, because of the unusual odor production, and it's not present when you go to a doctor, so the physician suggests you go see a therapist or someone with psychiatric training, and they in turn may say you're depressed or whatever, and give you psychotropic drugs, many of which have sulfur and nitrogen, the elements sulfur or nitrogen in them. And these drugs are handled in your body, in your liver, by the same oxidation system that oxidizes trimethylamine to trimethylamine oxide. So you're actually, if you put on a psychotropic drug and you have undiagnosed TMAU, that may actually make your symptoms worse. So this is something I've pointed out to any clinician I've talked to, but it remains to be seen how careful everybody listens to this. But most of our patients appear to lead normal lives and work at a variety of professions. The main problems, as everyone agrees, are psychosocial, and it's often tied to individual personality. I like to tell lots of people that many of my patients are physicians and clinicians, salespeople, lawyers, so they deal with the public all the time. You can control the odor, and so you don't have to be afraid. To summarize what I've talked about, our results demonstrate that there is a need to screen for TMAU with an objective analytical test and not rely on olfactory reports or how people look at you on the subway or public transportation system. Get diagnosed is the best thing to do. Only about 10% of our TMAU patients had the classic fish-like presentation either before, during, or after their choline challenge. Patients may not be aware of when their own odor is present or the intensity. So it's always good to have a friend who knows what your problem is and who you can rely on, whose judgment you trust, to tell you when the odor is present. And certainly having an odor buddy, as I call them, is absolutely critical anytime you're trying anything to control symptoms, because they can give you a fair assessment of how you smell at a social distance. Don't stick your hand on your clothing or in your stick your hands in into your nose. Those are intimate distances, and it doesn't mean somebody 5 feet away from you can smell that. Most of our referred patients had a very similar history. Many do not smell badly at all, and we do, as I said, do organoleptic evaluation of their breath, their underarms, their clothing, their upper back. Very few patients have high level of odor-producing bacteria in their axilla relative to normal people. So most TMAU people or TMAU negative people that we've seen don't have very strong underarm odor. I can usually outstink most of these folks if I go through the same protocol we put people through. We have worked with short-term and longer-term amelioration of symptoms, but there has been no good double-blind study of anything we have tried or anybody else has tried. We've tried it on a few patients. Some of this has been published in work I've done with John Cashman in terms of amelioration of symptoms, and Dr. Yamazaki from Japan. Continuing on, both our TMAU and non-TMAU patients may also have bad breath, and we'll be writing about more on this as time goes on, but about 57% of our TMAU positive patients have what I call a double whammy, so they also have bad breath and they have TMAU also. And about 60% of the non-TMAU positive patients have breath odor noticeable at social distance caused by what are volatile sulfur compounds emanating from the oral cavity. And this has nothing to do with TMAU. It's a bacterial condition that exists on the back upper part of your tongue. That's a whole separate lecture. So female patients either appear to be more affected by TMAU or appear in greater numbers to physicians or to us for relief of symptoms. Many of these in our population have been African American women. Decreased FMO3 efficiency due to complications. So there may be other reasons why TMAU rears its head in a person. Viral infections or drug intake may affect the liver or the FMO3 efficacy. So TMAU symptoms may appear after this or maybe appear sporadically and then go away because of situations like this. Certainly we need funding for more follow-up and in-depth study of symptoms and treatment regimens. And are there other genetic disorders that produce odor? Possibly, but we haven't been able to isolate and identify many of these as yet. There is something out there I know that everybody knows about, dimethylglycinuria. It's only been reported in one patient and documented in one patient so far. So there has been no widespread testing for this disorder. So that's the end of my talking. I'll turn it over to Dr. Reed. Danny?
Hello, everybody. This is Dr. Reed. I'm going to walk you through some of the basics of TMAU genetics. And to bring everybody and pull all these strands together, I'd like to start with reviewing just some of the basic biochemistry and molecular biology of trimethylaminuria. As we've heard from Dr. Fennessey and Dr. Preti, people ingest choline, they eat it, and through the action of bacteria in the gut, trimethylamine is produced. Now that trimethylamine is taken up by the human liver and it's processed by an enzyme that humans make called FMO3. This TMA is oxidized to TMA oxide, and that's odorless. However, when the FMO3, this particular enzyme, doesn't function at all or functions imperfectly, there's a buildup of TMA which gives the characteristic odor of trimethylaminuria. So, now what does it mean when we say that the FMO3 gene and its associated enzyme product in the liver, it doesn't work properly? So what I'm going to do here is sort of walk you through this gene structure here. This is a little snapshot of the FMO3 gene, and remember that genes are made up of DNA in your chromosomes, and there's a blueprint in every cell, and that from that DNA blueprint, the FMO3 enzyme, the protein that does the work in the liver, is made. Now you'll see that there's a whole bunch of letters and numbers here, and this information is building on some information shared in the second webcast of this series. And what you should understand from this is that the DNA sequence can be a little different from person to person, and sometimes these individual nucleotide differences really don't make much difference at all in how a gene might function. And sometimes they make a tremendous difference. So a single base pair change might lead to a protein that doesn't function at all. And that's often the case for people that have trimethylaminuria. They have a mutation or a genetic variant in this gene that completely renders this gene unable to function at all. But many patients have genetic variations that land somewhere in the middle. There's sort of a gray area, so that the FMO3 gene and its protein product works imperfectly. And so that's what I have to say about that. The thing to bear in mind is that trimethylamine is something that is produced naturally by human bodies, and that actually all otherwise healthy people have a very small amount of trimethylamine concentrated in their urine. And actually there's been studies that have looked at trimethylamine concentration in otherwise healthy people, and there's a big range of variation among these healthy people. So we're sort of thinking more about trimethylamine in the body as a quantitative trait, and not so much as a black and white disease or not disease kind of idea. And when we do that, we open ourselves to the possibility of finding other genes that might modify the amount of trimethylamine that the body can make. So one such gene, I'm going to walk you through this. So this is kind of complicated, but let me just walk you through this experiment. So this is an experiment which took otherwise healthy people and had them pee in cups, and the trimethylamine as well as many other metabolites were measured in the urine. And then the investigators looked at the DNA of all of these subjects very comprehensively. And as I said, people vary in their DNA structure. And so what these investigators did is observe that everyone in this population who tended to have very high levels of trimethylamine in their urine tended to share a little region of their DNA. I'm just trying to grab the pointer here. A little region of their DNA in common. So these, what look like gray lines with black dots at the top of them, these are indices of statistical significance. So how you should interpret this is to mean that in these particular regions here, this is a piece of DNA running along here, and in this particular region of the DNA here, people who had high levels of trimethylamine in their urine tended to share genetic material, the particular pattern of DNA variation, more often than you'd expect by chance. Now, this, as I said, is a piece of DNA, and above this you can see that there's little sort of word-like things here, and these are the actual genes that are associated with this little snippet of DNA. Now, recall that there are about 22,000 genes in the human genome, but in this particular interesting region here, this gene, PyroxD2, seems to be right on top of this particular genetic region. Now, we don't know much about this particular gene. It looks to be to code for an enzyme that looks to be capable of oxidizing metabolites, so it's not a far-fetched idea that it might participate quite directly in oxidizing trimethylamine. So, and that would explain, you know, the variation in trimethylamine and TMAO in the urine and blood of these particular people. So what you should take away from this is that FMO3 is a gene that you may be familiar with as causal of TMAU, but that there may be other genes that modify or work together with this particular to cause this particular trait. So I just wanted to introduce you to the idea of what we call in genetics epistasis, and it's just the simple idea that two or more genes may interact. So for instance, if there's two weak genes, so a weak version of FMO3 and a weak copy of PyROXD2 together may not have sufficient functional ability to actually oxidize TMA effectively and may lead to excess TMA production. So, which raises an interesting question, which is, are all cases of people that have TMAU due to FMO3? And so we have started a project which is called the Exome Sequencing Project. I'll just say a word about this word exome. You're familiar with DNA sequence. We have 22 human autosomes, and about 5 or 10% of that, our DNA sequence, actually codes for proteins that are the things that do work in our body. And the other parts of that are probably regulatory and don't directly code for proteins. So the exome is the protein coding region of the genome. And it's, of course, very important because when there are genetic changes in the parts that make the protein, it can have a very devastating effect on the ability of that protein to do its job in the body. So currently we have a project ongoing where we have 23 patients that have a range of TMAU symptoms, but these are not explained by traditional and known variation in the FMO3 gene, so they're sort of mystery patients. And what we're doing is sequencing the exome and trying to shift through all the variation we see to see if we can find additional genes that may be causing these particular set of symptoms. So this is work that's ongoing, and we're busy in the lab as I speak working on this project. So I just wanted to bring you back to a step in the cascade of TMAU that we talked about. And I want to focus your attention right now here on bacterial action. So I've told you that when we ingest choline, bacteria in the gut act on this choline to produce TMA. And we know that because we can take at least some bacteria out of the body and put them in a dish and feed them choline, and we can see that in a test tube or a dish that TMA is produced. So the idea that particular types of bacteria produce TMA is a well accepted and well understood idea. But up until this year, we didn't actually know how this was accomplished. What were the actual enzymatic steps? And just very recently, we've learned much more about this process. And so I want to walk you through this slide because it really represents a new, a new breakthrough in TMA research, and it's also a very clever and creative idea by these chemists from Harvard. So they reasoned here, here we have choline, and it's being metabolized by bacteria in the gut to TMA, and then the downstream TMAO, which is, this is the odorless entity, and this is the odor-causing entity. But the exact nature of this step was not known. So what these investigators did is they said, well, we don't know, this is a missing piece for us, but let's look for something that's similar that we do know. And so they focused on this reaction here, ethanolamine, where the enzyme here, this lyase, was known to oxidize and metabolize this product. And so the investigators searched through bacteria, and this is just the name of the particular bacteria, and bacteria are stored in a bacteria library, and this is just the sort of call number of this bacteria. And they found 4 enzymes that were very similar to this pathway that was known. And so they discovered a new set of enzymes here, choline trimethylamine lyase, and they've nicknamed this CUT-C and CUT-D, and the CUT stands for choline utilization. So that this enzyme, which lives in a bacteria, which lives in your gut, is now known. And the reason that this is important to patients is that it leaves open the possibility now that we can directly measure the amount of bacteria and the amount of CUT enzymes in each individual person's gut to give a sort of idea of why some people might be more severely affected than others, or why might different foods change the gut bacteria composition in a way that might favor an increased production of trimethylamine, or it might reduce production of trimethylamine. And it also leaves open a very exciting possibility, since now that these enzymes are better understood, that there may be small molecule inhibitors of these enzymes that might damp down the production of TMA in the gut in the early stages, which might lead to some symptom relief. So I wanted to just leave you with an interesting and provocative piece of data. So this is a study. I'm just going to describe to you what this data means, and then I'm going to tell you what I think the interesting implications are. So as I said before, it's now possible for scientists to take the urine of human individuals and animals and screen for a broad variety of metabolites, not just TMA or TMAO. And so these large-scale screens are going on right now. So people are just screening the urine of human subjects and animal subjects, and then looking to see how different metabolites in the urine match up against different kinds of diseases. It's kind of a look and see rather than a hypothesis-driven kind of approach. And here's one very interesting recent finding. So these investigators found that TMAO, and remember this is the oxidized form, so this is the the form that TMA patients typically don't produce very much. They found that this high levels of this oxidized form is associated with atherosclerotic plaques. Now you'll remember that this is atherosclerotic plaques are associated with arterial disease and heart disease. So it leaves open the very provocative idea that maybe there might be some health, I don't want to say exactly health benefits, but it could be the case that people who are unable to produce this particular compound, like those that have trimethylamine, may be at less risk for heart disease. So you remember that Dr. Preti showed a list of diseases that may be associated with TMAU, but that heart disease wasn't on that list. So this is obviously a very new and unusual idea, and obviously something where we need much more testing, but it's something I think that it's really worth pursuing and could be quite interesting. So I just want to wrap up to say there are many known genetic variants in the FMO3 gene that lead to frank cases of trimethylaminuria. And it's also good to remember that genetic variation can occur on a spectrum. So some types of genetic variation leave a gene dead cold. It just can't function at all. But there's a significant gray area. And when gene variation is in this gray area, it might be more easy to push that function around by things like diet and exercise and stress and menstrual cycle and so forth. We also now know that there's another gene that, at least in otherwise healthy populations, pyroxD2, seems to be associated with trimethylamine in the urine. And how these genes might work together is still an open question, but it leaves the idea that there might be important to genotype both genes. We also have a project going on to do exome sequencing to look to see, without any prior prejudice, what kind of variation we might find in patients who seem to have very serious symptoms, but we can't find particular mutations in the more classically known genes. We've also, investigators have made big strides in understanding the amount of bacteria in the gut that, and the enzymes in gut bacteria that actually convert choline to TMA, and this leaves open a big possibility that new therapeutics might be developed. And finally, I shared with you a provocative and very some food for thought that there actually may be some benefits to the inability to convert TMA to TMAO, and of course that's an open area of research. So I just wanted to say that I'm a new person in the TMAU field, and it's been great to be invited to be part of this particular research team, and I'm also very pleased to be with you here today. I just direct you, if you're interested in more information, to our website, which is www.monell.org. That's www.monell.org, and just take a moment to say thanks to everybody.
Okay, thank you very much, Dr. Reed. This has been a very interesting webinar with three presenters that made some really clear summaries here, and we appreciate that. I wonder if you guys might have a few minutes for questions.
Yes, I will be glad to answer questions.
Okay, so we can take questions either by microphone. If people want to enable their microphone, they can ask a question, or they can just type there in the chat box. And I think we actually already had a question from guest 42. Since so many people have the symptoms of TMAU but test negative, is there any plan to revisit the criteria for the test, such as testing blood, DNA, or stool?
If you haven't been tested by the choline challenge test, you haven't really been tested. If you haven't been tested by if you're a female and you didn't have the test during menses, I would suggest you repeat it and have it done during menses, because many women are primarily symptomatic at menses time, and that's when the test will be of most benefit, in quotes, to you. That's what we recommend to all females, that they do the testing, and that's in our very detailed letter of instruction. Number two, since Danny and I work at a place that specializes in odor analysis, and we have a lot of our colleagues are sensory psychologists who study odor perception and how people describe odors, I can tell you that unless you have a variety of people evaluate you, you may not smell fecal to me. To me, your what you perceive as fecal may smell like sulfurous, or like swamp gas, or like sewer gas. So I often associate fecal odor with sulfur compounds, which are the principal components within fecal material, because I've analyzed swine farms and I've analyzed human fecal matter, so I'm pretty familiar with those odors. So you can be tested. I don't think there's we're barely getting enough money to do what we do, so revisiting criteria to look at A, B, and C, I don't think so. Danny's work may reveal other things that are going on, but unless you've really been evaluated by the choline challenge test at the time of menses, when your symptoms should be maximal, unless you've had other parts of your body looked at analytically with the tools that we have available in our lab, you may have been either misdiagnosed, or you may be maybe perhaps you don't smell as bad as you think you do. But regardless, I would certainly suggest redoing the choline challenge test at the time of menses if you've tested negative once. Okay, I see that guest 42 took the test for me 3 days before menses and tested negative. I'm also allergic to sulfur medications. I don't, have you been here? If you haven't been here, then I don't know what else, I don't know how, we haven't evaluated you in vivo.
There's also some questions there on the discussion notes panel. Can you see those? Starting with, are any gastroenterologists interested in researching this, since it is related to gut bacteria?
I haven't run across any at Penn. I don't know if Paul has run across any at Colorado. Paul?
Paul, are you there?
Yeah, I'm here, yeah. I'm sorry, I just was clicking the wrong icon. Yeah, we have a whole group of GI docs here, and none of them have really shown any interest in this. I think because the work that Dr. Reed was talking about is really very recent. And so I think with that information, with the cut-in signs, there may be more people who are going to get interested in actually looking at more details of the gut bacteria. I mean, when you get down to it, these are, again, estimates. People talk about not millions, but billions of bacteria living in the gut. And so it's going to be an interesting thing to sort out ones that are TMA production oriented and ones that are, don't have any effect on TMA. So it's not an easy.
Rob, this is George. Do you want to continue to host this, or do you want me to just take questions as they pop up? Because there's a lot of them popping up.
Yeah, there's a lot of questions popping up, George. And also, can you see the questions box now on?
Yes, I can see it.
On your
I can see it.
Okay, yeah.
Yes.
So those are the questions that I've copied up until now from the chat. So if you want to take those as you can, or if you even feel comfortable answering them all, yeah, just take those.
Okay, I'll start with the top one. Maybe Paul can chime in too. Is TMAU included in medical journals? Well, it's not included in medical journals, but if you go to PubMed, which is the public access to the National Library of Medicine, and put in trimethylaminuria, you'll get all this peer review articles that have been published, abstracts of them, that have been published about trimethylaminuria, or if it's been included in the keywords that the authors put in. So, or if you put my name in or Fennessey's name in, you'll see the articles that we have published, not only on trimethylaminuria, but on all kinds of other things that we've worked on. So there are ways of access. If it's a keyword in a paper, it will be in the medical literature, so to speak. Paul, do you want to add anything to that?
Not really. I think that's probably the best source of information. The problem is you have to get to the secret word TMA or TMAU to get that out. And that's the problem. We need, we've been working with some of the people that are writing now the newer biochemical journals and medical journals, trying to get at least references into them about trimethylaminuria.
And yes, I have heard about the human microbiome sequencing project that's ongoing at NIH. The work that Dr. Reed talked about is from the Department of Chemistry at Harvard. But don't write to that young lady. She probably doesn't know much about trimethylaminuria. She just studies the chemistry of certain enzyme systems. So people are working on this, but it's not an easy problem. It's very, very complex. Like Paul said, you have billions and billions of bacteria in your large intestines, and those bacteria may also infect, in females, the vaginal area. So if your odor is prominently from your vaginal area, these cut enzyme-containing bacteria are anaerobic, and they can live in the vaginal barrel also. If they become overwhelmed, if they become the dominant population there, then you will have more and more odor symptoms from your genital region. So it's not an easy situation, and it takes time to accumulate knowledge. And knowledge, the mother's milk of knowledge is money. And without money, we don't do research, and the progress is very slow. Okay, do I think juicing...
So
go ahead, Paul, I'm sorry.
That's good. Thank you, Juicy One.
Juicing vegetables would help with odors. Well, don't juice onions in them, because they'll give you oniony breath. Onion and garlic are good for you, but they will retain odor on your breath, not many other places. So juicing vegetables, yeah, I take a lot of vitamin supplements. I don't take the time to juice vegetables. I eat raw vegetables when I can, but that's up to you. I don't know if anyone's done any studies on things like that. Why can't families smell it in many cases?
Let me grab on to the next.
Go ahead.
Yeah, so this is an interesting one, because I think a number of patients with trimethylamine excess can't smell the disorder, the TMA themselves. They only know that they have a problem from, as George would call it, a smelly buddy, who tells them you're really reeking today. And it, I think, becomes, what happens is the trimethylamine is in so much of the system that the your nasal pharynx cannot detect the trimethylamine because it's saturated with trimethylamine. It never does. It doesn't change one way or the other because there's so much there. And it may be if your family is exposed to trimethylamine, a lot of it, they became sort of, what do you say, overdosed and muted to being able to detect the smell. So it's an interesting sort of aside to the disorder. George?
No, well, yes, you can get, you do become adapted to odors in your environment. So family members might, if you're coming in from a day of work of being outside the home, family members may be able to perceive it at that time if it's present. So it's an iffy question as to how good a family member might be at detecting trimethylaminuria mediated odors. Certainly, if you have bad breath, your spouse is probably the best one to ask, because in the morning when you first wake up, that's when bad breath is present at its worst. So that ad they used to be on television about morning breath, that was probably one of the truest ads on television, because that's been documented in real studies, both organoleptically and with analytical instrumentation. At menopause, do odors get worse? We have not done any type of study on that. There's just not been any. I mean, our group data suggests that menopause and women of reproductive age produce about the same amount of trimethylamine oxide. It's when women are menstruating that the odor can actually get worse. So the answer is we're not really sure, but the data we have on hand suggests no, it's going to be about the same.
So George, you've done some stuff on pre and pro antibiotics for, not antibiotics, but treatment for trimethylamine. You want to?
Many of the people who come to me have tried this. I mean, but we haven't done any systematic study of taking people off and on a probiotic. I tell people to take it. It can't hurt, but remember that the fact that regardless of what you do, you can't sterilize or totally change the bacteria around in your intestines. You may make the predominant ones less effective at producing trimethylamine, but they're still going to be there. So try it. If it works for you, that's fine, but always have an odor buddy. I mean, you can't go wrong by having someone you know and trust evaluate what's going on for you. Might other FMO3 substrates cause the odors? Paul, you want to start off on that?
Well, yeah, I don't know the answer to that. I mean, I do know that that dimethylamine and other kinds of compounds do cause odor, and so I would, just to stretch a guess, I would say the answer to this is yes, that there are other substrates that are maybe not handled by FMO3, since FMO3 is usually a nitrogen oxidation. One of its processes is a nitrogen oxidation biochemistry. There may be other nitrogen-like compounds that have strange odors associated with them, that if you have a defect in FMO3 or related enzymes, that you actually generate odor from that. But I don't know that, again, that there's been anything really systematically done with that.
Is what?
Is Danny still around?
No, Danny had to leave.
Is Danny still around?
Yeah, she had to leave.
I don't see her on our...
Yeah, that would be a
okay.
she might be better able to add to that. I think I've already pontificated on the human microbiome sequencing project.
So there's one down there that's kind of interesting, George. One comment about I had the choline challenge test. I don't know when that was done at the Colorado lab, and it was deemed negative, but I had a blood test genetically,
well,
and
you didn't
it
have it
showed
with me
positive
because
TMAU.
we generally don't test people genetically for if they've tested negative through the choline challenge. So I'm not sure who that person, I don't know who that person is, but he or she should contact me directly and illuminate me.
Right. I think that would be good. I think she should talk to either of us or send us data.
Yeah, me.
Because I'd be curious to follow up on that too.
My doctor thinks I have a methylation problem. Well, how does he know that? He or she know that? A methylation problem, you might be in, I don't know what in God's name they're talking about, methylation problem. Do you, Paul?
No, I don't. I mean, that would be...
You have a major problem if you were not able to methylate certain things.
I wouldn't go there. I just don't understand.
Yeah, so I don't know
You'd be in real trouble.
what your doctor meant by that. So sorry. If you can get a better definition. Go ahead.
Okay, let me just go to the bottom one. It's me who has that reaction, namely when I'm with a TMA patient, my eyes run liquid tears. And it's not an immune response, it's an irritant. It's like any time you go into a place where there's some kind of a gas or vapor that burns your eyes, the tears come. So for some reason or other, trimethylamine causes my eyes to water like crazy. It almost looks like I'm crying.
That's a trigeminal
when I'm
response.
talking to a
The
patient.
trigeminal nerves are the nerves activated during irritation. Same type of nerve system that that's activated when you take, albeit different receptors, hot spicing and things like that. But the trimethylamine levels would have to rise to a fairly high concentration for you to, you smell trimethylamine at one level, and about 100 times to 1,000 times higher, it would become an irritant. So if your eyes have become irritated by TMA levels in the presence of a patient, they were producing a hell of a lot of trimethylamine at that time. Okay, now that was Dr. Fennessey. Next question: do you think that that saying TMAU treatment with low choline is a misconception? No, I don't. When sulfur tyramine gives you trimethylamine also. Phenylalanine are amongst other compounds in the diet contributing to the odor. Not necessarily so. Sulfur might. Phenylalanine, there are only certain foods that have a lot of phenylamine, and the Alliaceae and broccoli-like vegetables contain a lot of sulfur, and those have been shown to exacerbate symptoms. I think John Cashman did a study in this area a while ago. So yeah, you should try to avoid certain types of vegetables when you do have trimethylaminuria, but you can't avoid everything. You have to take everything in moderation and see what works best for you.
Yeah, I would just chime in here, and I don't know whether Sandy's got a microphone anywhere or not. I see she's on making comments. But my point would be that this is where the TMA network and webinars like this come in to be very handy, because there are some foodstuffs that exacerbate this disorder, and we haven't touched every single one of them for sure. And so having people who have tried and tested other possible food regimes, it becomes important to pay attention to their experiences. And although it's anecdotal, I would say it's better to be a little safe and pay attention to them and see if you have an odor buddy, as George calls them, to try either taking or not taking some of those foods and seeing if you have a exacerbation or a reduction in odor.
Right.
It's worthwhile paying attention to that group.
Activity TMAU. Do you feel that certain medications can make people toxic from a built one due to decreased FMO3? I don't know if it makes you toxic, but I think, as I said, certain medications, because of their chemical structure, may compete for the FMO3 enzyme activity and therefore cause you to not oxidize trimethylamine and other compounds as efficiently. So I tend to avoid the word toxic because it just has a different type of connotation to me, but it can make you feel ill or it can make you smell. So if you have trimethylaminuria and you're taking medications, certainly be aware that you may produce more odor under those conditions. And once again, this is where an odor buddy may help you a lot.
Yeah, right. That's very true. So the next one, what percent, I assume it's what percent of TMAO to TMA makes us sets a diagnosis of yes or no for TMAU. And we've used a cutoff of like 93 or 94% TMAO over TMA itself. So I don't know whether that's a really good systematic number, but it's the number we're using at this point in time. Anything where the TMA gets high enough that the ratio drops below 93%, we consider to be a positive.
And but in my clinical experience of seeing
patient.
people, those folks with a percent above 70 or 80%, if they're in this 75 to 90% range, that's, to me, those are very mild, a mild case, and I have not perceived an odor in the presence of people like that, trimethylamine type odor in the presence of people like that. Generally, when people smell of fish, they have a very severe case and percentages of TMAO of less than 30%. Can TMA cause? Yeah. Yeah, there's two questions in here about irritancy. TMAU cause people to cough. Is it being an irritant the reason why nearby people sneeze a lot? I'm going to challenge the person who wrote this, either one of these, to demonstrate to me that come in here and stink your best and see if you can make anybody I bring into lab sneeze or cough. You would have to produce a hell of a lot of material, to give those irritation symptoms. Can TMAU cause the person who has TMAU to cough? I don't think so, because I go to at least one clinician in the city who has a severe case of TMAU, and I'm glad he doesn't cough when he's doing his exams on me. Is it an irritant to other people why nearby people sneeze a lot? Well, we have never had a person here in lab who has made us sneeze, and we've been able to smell them. So once again, you have to produce a lot of odor to get it up to irritancy level. Occasionally you may have a person who's very sensitive to irritants, like maybe Paul is to this in his eyes, but in general, you will not produce enough unless you've taken a hell of a lot of choline and have very low enzyme efficiency to make people sneeze.
So let's look at the next question. This one about negative FMO3 sequencing. I think this one is backwards in a sense. That yes, you can have a negative FMO3 sequencing test or gene test, but still have TMAU, and that's what Danny was talking about, in that there are other enzymes that actually either regulate or exacerbate or what should I say, cause the higher production of TMA. So just because your FMO3 enzyme is seen to be quote, quote, normal, doesn't mean that you can't have trimethylaminuria. I think we haven't gotten to the bottom of all the possible causes for this, including some cofactors that work with this enzyme. And as George has pointed out, sometimes drugs can just overcome the capability of the enzyme to work, and therefore you end up with a smell. So don't get a sequencing done first, get a biochemical test done,
there's a question
and then start to look at the sequence.
About bacterial overgrowth. If I can just, before we go to the other one, you already have multiple billions of bacteria. In fact, I think I saw an estimate once or twice that there are a couple of pounds of bacteria in your large intestine. So I can't imagine what the hell an overgrowth means. This is some sort of internet phrase I've seen tossed around a lot. You generally don't have bacteria in your small intestine and in your stomach. Most of them are way down there in the lower gut. So I don't think TMAU is not going to, symptoms are not going to arise by bacterial overgrowth, because if you go to, I don't think anyone can define what a bacterial overgrowth means when you already have billions and billions of them in your large intestines. So if someone can tell me what that means, maybe I can build a better argument for or against that, the answer to this question. That's different.
So George, there is a clinical condition in which peristalsis slows down, and in which bacteria actually do grow into the small intestines. And I don't know about whether or not that produces or exacerbates trimethylaminuria, but there is a small percent of the population that our GI doctors deal with that has to do with bacterial overgrowth in the small intestines. I just don't think that anybody's looked at the TMAU piece of that.
But how sick are you when you have that?
It's a threatening clinical problem.
Yeah. Okay. That's an answer then. If you're walking around and concerned about odor, then you probably don't have bacterial overgrowth in quotes in your small intestine, and your stomach. Unless you have that Helicobacter pylori growing in your stomach, in which case you may have ulcers. Bacteria generally don't hang out in the stomach. The conditions are just not right for it. Can't you find some odor absorber or antioxidant or something that takes the odor or the gas away?
Okay.
Sure, you got the money to pay for a study where we can look at a multiple set of odor absorbers and things that are approved for human use. Antioxidants, you can go to any store and buy lots of antioxidants. There's I don't know which ones will take odor away though, but a lot of them will oxidize. They don't work in the liver though. They work by different mechanisms to oxidize different things, and it's generally not small molecules like trimethylamine that they're working on. Someone snuck a genetic question in without Danny being here.
Okay, I don't know
I have no clue what
266782 is.
What change that is.
Right.
Yes, excellent. Well,
I think, try to get in touch with Danny. That's the right thing. Right.
Part of the protocol. Well, we
That's all right.
recommend a
Lost
lot
the
of,
second
we
call.
recommend low choline intake, and again, there, as I'm sure Sandy Gordon and others have preached on their website and in their discussion groups, that there is no diet per se. I give people a list of foods with choline in them, and I recommend that they take it to a registered dietitian to formulate a diet for them that is low in choline, albeit still nutritious for them. In concert with that, I tell them to take a probiotic if they wish, but to first start off with a 10 to 14-day course of an antimicrobial that will blow away many of the gut bacteria, then start taking the probiotic, start on the low choline intake, and we also recommend certain over-the-counter compounds that will interact with trimethylamine at the gut level. So we do all those things to try to, and we ask people for feedback. We don't have any mechanism for follow-up. The feedback I've gotten is generally from the most severe patients, and they tell me that when they stick to the things I've told them, things work. And we are working with a lot of parents of younger people in high school where social implications are very acute. So I know what we're doing is working because, or certainly helping, because many of the kids that are following my instructions are getting by without any social consequences.
Okay, let me touch the liver transplant. There is really no data about that, because TMAU is such a rare disease that having trimethylaminuria and having a liver transplant, I don't even think you get in line for a liver transplant since there are so many people demanding them because they have complete liver failure. But I would guess that if you did have a liver transplant, you would mitigate the odor production, at least as long as the liver was able to continue to produce FMO3 at normal levels. And it does give us a hint of where treatment might really come for this disorder, and that is a lot of the people at Stanford now are looking at stem cells and particularly trying to direct stem cells to grow in a certain direction, like muscle, or liver, or kidney, etc., etc. And if they can ever figure out how to direct a pluripotent stem cell into liver, you might actually be able to inject it and get it to grow in that organ. Those are real speculatory kinds of things, but it's really, I think, the real hope for any kind of ultimate cure for this disorder would be from the stem cell program.
I assume they mean someone with severe case of trimethylaminuria have no odor. If you are that person, give me a call. I'd like to talk to you because it seems incongruous at first glance. What do you think, Paul? Be no odor, yes.
I agree. I think it would be difficult to have high levels of trimethylamine and no odor. I mean, you may not be able to spend the people around could.
Right.
And the antibiotic treatment, I think, it's true it works, but no one in their right mind, I think, would recommend it, only because the antibiotics, actually, the way it works is it kills these millions, thousands of bacteria in the large intestine, and it does reduce the production of TMA, but the side effects are chronic diarrhea and, all kinds of other absorptive problems. These bacteria are there for a reason, and you don't want to have, trimethylamine
I don't know
for
what
having
alpha-1
the sports for the rest of your
antitrypsin
life.
deficiency is. Do you, Paul?
It's not a good trade-off.
So since we don't know what it is, we can't answer.
Hey, you two both did amazing at that battery of questions, and
So
we really appreciate
we
you staying on so long after your presentation to answer those questions. And I know people can continue to get in touch with you when they have questions, or
Sure.
visit the Monell website, or like you said to a few people that raised issues, call your lab.
Is this Rob?
Yeah, this is Rob.
Okay, looking at the chat list, it looks like people are still furiously typing in, and Sandy is hurling back answers at a variety of people, along with Maria de la Torre. I don't know if you have the capacity or desire to do it, but if you want to summarize some of the questions we didn't answer, and send them to me, or I don't know if Paul's game, but I'll be willing to answer some. It just won't happen right away. Because I'm getting dozens of emails a week anyway from people, and it usually takes me a while to answer everything.
Rob,
Yeah.
Rob, if you would just take the list from the chat thing and just copy it and mail it to us,
Right.
both of us, and let us work on giving answers to them. That's the easiest way, I
Yes,
think.
sounds good.
And that way no one gets shafted in the
that
sense
sounds
of
good.
not getting their
I'll
question
collect
answered.
the last couple questions
It-
that
it-
were kind
but
of
us, George,
outstanding there.
will take a while.
And hopefully everybody got a chance to ask
That-
their questions.
Should we
Thank you
sign
very
off?
much for your time today on this Friday, the first day of February, where I think you really explained some things in very friendly terms for all of us, and then answered everyone's questions.
Okay,
So,
very good.
yeah, we'll have the recording of this ready on Monday to show to you first, and then we'll be able to share it with the people who are here, and also with the people who couldn't make it here today. If that ends up being okay. So have a nice weekend, everyone.
Okay, you too. Bye-bye now.